Welcome, focused learners. I am absolutely thrilled to have you here for this explainer. Today, we're looking at something truly fascinating. When you look at a massive, thriving field of wheat or incredibly resilient, drought-resistant corn, you probably just see the triumph of nature and biology. But the reality? The future of our food is actually written in complex mathematical formulas. Behind every modern supercrop is an invisible engine of data. Okay, let's dive into this. What we're really talking about here is biostatistics. Formally, in plant breeding, statistics is the science of collection, classification, analysis, and interpretation of numerical data. But I want you to think about it differently. This isn't just a collection of numbers in some dusty textbook. This is the critical toolkit that scientists use to effectively hack plant genetics, building better, stronger, and more nutritious plants for our growing world. To understand exactly how scientists pull this off, we're going to follow the incredible journey of a crop. We'll start with the math behind better plants, figure out how to find the average crop, and then measure plant variability. From there, we'll calculate the odds of good genetics, test our breeding hypotheses, and finally predict future crop yields. It's an amazing progression from a simple field observation all the way to complex predictive modeling. Let's get to it. Let's kick things off with Section 1, the math behind better plants. Think of the progression of biostatistics a lot like leveling up in a complex video game, right? You can't fight the final boss without mastering the basic tutorials first. Mather and Jinks outlined 3 degrees of statistics here. Level 1, or first-degree statistics, deals with means. These are robust, highly reliable, and delightfully simple to calculate. You absolutely must master these simple averages before you unlock Level 2, second-degree statistics. This is where we calculate variances. It's a much more difficult calculation, it's less statistically robust, and it requires way larger sample sizes to get precise. Finally, you reach the third degree, looking at skewness and kurtosis, which scientists use for fitting frequency curves. It's a clear, sequential path. Moving right along to Section 2, finding the average crop. So imagine a breeder walks into a massive field of thousands of distinct plants. How do they figure out what a normal plant even looks like? Well, they do this by measuring central tendency, and different measures solve very different breeding problems. The arithmetic mean is your go-to for estimating average performance across the board, things like mean yield or plant height. But what if you're testing for survival in really harsh conditions? That's where the median steps in to save the day. When evaluating extreme values, like extraordinary drought or salinity tolerance, the median doesn't get skewed by a few outlier plants that happen to survive by pure chance. And finally, we have the mode, which is perfect for identifying the most common phenotypes, visual traits like a specific flower color or categorical disease resistance scores. Which brings us to Section 3, measuring plant variability. Now, what's really interesting about this slide is once we know what the average plant looks like, the next logical question is, how different is everything else? Just look at this beautiful hand-drawn curve we have here. This is the normal distribution. It's perfectly symmetrical, bell-shaped, with asymptotic tails stretching off infinitely in both directions. The amazing thing about nature is that most of the quantitative traits we truly care about in agriculture, like total crop yield, plant height, and days to maturity, naturally follow this highly predictable bell shape. Understanding this curve tells breeders exactly where the superior outlier genetics are hiding. But to actually measure that curve accurately, we really need to understand a brilliant dichotomy from our sources: standard deviation versus standard error. I know, they sound super similar, but they do completely different jobs. Standard deviation measures the physical spread of the data around the mean within just one specific sample. It tells you how varied your field is. Standard error, on the other hand, measures the precision and accuracy of the sample mean itself. So simply put, deviation is all about the spread of your plants, while error is about the accuracy of your math.
All right, on to Section 4, the odds of good genetics.
When you get right down to it, crossing 2 plants and hoping for a superior offspring is a lot like playing a high-stakes lottery. To win that lottery, scientists use probability distributions, like the ones neatly organized in this table. We've got discrete distributions, like the binomial, for fixed outcomes, say whether a plant is disease-resistant or not. But look closely at the Poisson distribution. This one relies on a constant rate and is used for describing super rare events occurring in a unit of space or time. In plant breeding, you'd use Poisson to predict incredibly rare 1 in a million genetic mutations or sudden disease outbreaks. It literally takes the total randomness of nature and puts a mathematical boundary right around it. Now we reach the really critical phase in Section 5, testing our breeding hypotheses. So, We've found our average, we've measured the spread, and we've played the genetic lottery to find a supposedly superior plant. But here is the ultimate question. Is this new crop actually better, or was the soil just luckier? I mean, this is the million-dollar question that keeps agricultural scientists up at night. Is this new crop genuinely, genetically better, or did the soil in that specific patch of the field just happen to get a little more water and nutrients? Without a solid way to answer this, agriculture is just guesswork. To definitively answer that question, we enter the battleground of significance testing. Scientists evaluate two competing ideas at a chosen level of statistical significance. On one side, we have the null hypothesis, the assumption of no difference. It basically states that your new plant isn't special at all. Any variation you see is just random experimental error. And on the other side is the alternative hypothesis, the hypothesis of difference, which confidently claims your plant is definitively genetically superior. To declare a winner between those two hypotheses, breeders have an incredible statistical arsenal at their disposal. We have the t-test, brilliantly developed by Gossett for small sample sizes under 30. Then there's the z-test, championed by Fisher for large populations. There's the f-test to test the significance of difference between population variances. And of course, we can't forget the chi-square test, developed by Carl Pearson, which is used specifically for goodness of fit, to see if Mendelian genetic segregation ratios actually hold true in the field. And this brilliantly illustrates how all those individual tests ultimately feed into tools like this, the ANOVA table, or analysis of variance. Now, I know it looks incredibly complex, but it's really just a beautiful way to organize chaos. It neatly divides all the variation in a field into designated rows, variation from our treatments, variation from our replications, and the unavoidable experimental error. By comparing degrees of freedom and the sum of squares, breeders calculate the f-value. This gives them undeniable mathematical proof of whether their new plant variety is a true success or just a dud.
Which brings us to our final stage, Section 6, Predicting Future Crop Yields.
This is where we use predictive modeling to quite literally look into the agricultural future. Think about the massive scale of global farming for a second. You simply can't wait until the end of the season to guess your food supply. So how do we predict a massive harvest from just a few early plant traits? How do we take the leap from observing a plant's current early state in the field to accurately predicting its ultimate harvest yield months down the line? Well, the answer lies right here in regression analysis. Just look at this simple linear regression scatterplot graph. We map our independent variables on the x-axis. These are the early traits we can measure right now, like leaf area or plant height. We then draw a line of best fit to predict our dependent variable on the y-axis, the final yield. This functional relationship mathematically locks in our prediction, showing us exactly how a unit change in x impacts y. And I really want to emphasize the real-world stakes here. Regression isn't just a fun math exercise. It is the absolute foundation of modern agriculture. Breeders use it for the prediction of yield from yield components and to identify crucial traits for indirect selection. It drives stability analysis to see how a plant genotype interacts with entirely different environments. And incredibly, multiple regression serves as the very mathematical bedrock that empowers today's cutting-edge genomic selection and machine learning algorithms in global farming. To bring it all together, I love this quote derived from our source notes. Tests of hypothesis provide scientific validity to experiments and selection decisions supporting varietal releases. That right there is the ultimate takeaway. All of this biometrical analysis, the means, the variances, the t-tests, the regressions, it's all done so that when a new, potentially life-saving crop variety is released to the world, it is done with absolute, unshakable confidence. Which leaves us with a final thought to chew on. If statistics is truly the invisible engine powering modern plant breeding, what hidden math will be responsible for the food on your plate tomorrow? I challenge you to look at your next meal differently and appreciate the deep, unseen mathematical journey that brought it there. Thank you so much for joining me on this explainer. Keep asking those big questions, and I'll catch you next time.